Good afternoon. Imagine you're at a concert surrounded by the crescendo of a live orchestra. The drums thump loudly, the trumpets blaze brightly, and the violins soar with fervor. Now imagine there's a triangle player in the back, just a small subtle ding every now and then. You might barely notice it, yet without that triangle, the music wouldn't feel quite complete. Today I'm here to share with you something akin to that tiny triangle, the quiet, subtle sound which can change our understanding of disease detection. Specifically, I'll talk about a new way to listen to signals in our bodies using microRNAs as biochemical instruments. We need to focus on the blaring trumpets, those markers whose amounts change dramatically in a disease. But what about the triangles, the markers that shift just a little? Could those hold hidden clues that lead us to earlier and more accurate diagnoses? As it turns out, they can. Let's start with the basics. Our bodies are made up of tens of trillions of tiny living units called cells, each one like a microscopic city buzzing with activity. Inside every single cell is a master instruction manual, our DNA. This DNA isn't just a string of letters, it's a dynamic, living code that tells our cells how to function, grow, and heal. DNA, being the master copy, doesn't do the work directly. Instead, it sends out messages like emails from headquarters. These messages are called messenger RNA or mRNA. Think of mRNA as a courier that carries specific assembly instructions from the DNA to the cell's protein factories. Proteins are the tiny machines that do just about everything. They build cells and tissues, fight infections, carry oxygen, digest food, you name it. Proteins are the unsung heroes that keep us alive and thriving. This whole process, DNA to mRNA to protein, is known as the central dogma of biology. It's like the ultimate production line, blueprint, message, machine. And it's happening in every cell of our bodies, every second of every day. It's not just science. It's the symphony of life playing out inside all of us. But here's where the plot becomes even more interesting. There's a secret squad of molecules called microRNAs or mRNAs, and they're not your typical players. Unlike mRNA, they don't convey the construction instructions of anything themselves. Instead, they're like the backstage crew at a concert, controlling the spotlight, adjusting the volume, and deciding which performers, genes, get to shine and which ones stay quiet. These tiny molecules are master regulators, fine-tuning the symphony of gene expression with incredible precision. When everything's running smoothly, mRNAs help keep your body in perfect harmony. But in diseases like lung cancer, this balance goes haywire. Some mRNAs crank the volume way up on genes that should be silent, while others almost vanish, leaving important genes unheard. It's like a soundboard gone rogue, leading to chaos in the cellular concert. Understanding these microRNAs is like discovering a hidden control panel for your body's most vital systems. And scientists are now learning how to read and even rewrite these molecular messages to detect and fight disease and restore balance. It's cutting-edge molecular biology with the power to transform medicine. Scientists have traditionally honed in on those extreme changes because they're loud and easy to detect, much like the pounding drums and blazing trumpets in our orchestra analogy. Yet diseases have complex biochemical mechanisms. Imagine listening to a symphony and only paying attention to the loudest instruments. You'd miss out on the rich tapestry of melodies created by all the other players. That's essentially like ignoring mRNAs whose amounts change only moderately. Those median shift mRNAs are like the triangle, small but meaningful. In my study, I set out to test a simple but bold hypothesis. mRNAs with moderate changes in their quantities carry just as much diagnostic information as those with strong changes. To do this, I obtained data pertaining to over 1,500 lung cancer patients and more than 2,100 healthy individuals. Each person's blood sample provided the full profile of mRNAs. For each mRNA, I determined how much its level in lung cancer patients differed from healthy people, corresponding to its disease-specific dysregulation. Consequently, I categorized microRNAs into three groups based on how much their level shifted. Highly dysregulated, or HD, these had the biggest swings, either doubling or halving in abundance in sick patients. Medium dysregulated, MD, These changed by a more subtle amount, although they did not make it into the HD category, their levels changed up or down by at least 15%. Non-significant dysregulation, or ND, these barely budged at all, within plus or minus 10%. Conventional research picks the loudest signals, the HD group, and works with them. But I decided to build 3 separate decision models for diagnosis, one model for each group to see how well each group can distinguish between cancer and healthy samples. To create these diagnostic models, I turned to an approach from the field of machine learning. You can think of machine learning as instructing a computer to recognize patterns in large amounts of data. You show the computer many, many examples. This is a cancer sample. This is a healthy sample. And the computer gradually builds an understanding of which features best separate the 2 classes of samples. I used 2 machine learning techniques in this study: random forest classifiers and k-means clustering. Allow me to explain both with analogies. Let's start with random forest classifiers. You can think of it like a forest made up of many decision trees, each helping to make a prediction of the class, with cancer or healthy, of a sample. Imagine walking through a dense forest where each tree represents a quiz. Each tree with its trunk and branches correspond to a series of yes or no questions, such as, is the level of this specific mRNA higher than a certain value? And each branch split associated with a yes or no answer leads to another branch representing another question. By answering these questions and traversing the tree, each tree comes to its own conclusion, labeling the sampler as either with cancer or healthy. One decision tree can be a bit fickle. If it sees a slightly different sample, it might sway one way or the other. But what if you gather an entire forest of these trees, maybe 100 or more, and let each tree cast a vote? If most trees say cancer, the forest concludes cancer. If most say healthy, the forest goes with healthy. This majority vote system is called a random forest classifier, or RFC for short, and is remarkably more robust because it averages out the quirks of individual trees. The power of the random forest comes from combining the answers from many trees instead of relying on just one opinion. It takes a vote across all the trees to make a more accurate and reliable prediction. In my study, I developed 3 different random forests, one using only the highly dysregulated microRNAs, one using only the medium dysregulated microRNAs, and one using only the non-significant dysregulated ones. I carefully selected the 50 most informative microRNAs from each group, those that gave each forest the best chance to make accurate predictions. And the results? They were truly remarkable. The forest trained on highly dysregulated microRNAs achieved an accuracy of 99.7%. The one using medium dysregulated microRNAs achieved a comparable 99.2% accuracy. Even the forest using non-significant dysregulation microRNAs performed surprisingly well, with 92.1% accuracy. These findings highlight just how powerful and informative microRNA data can be, even when the signals aren't immediately obvious. Put simply, the medium dysregulation microRNAs, or triangles, perform nearly as well as the loud trumpets. They were not only capable of distinguishing cancer from healthy samples, but they did it with almost the same accuracy as the HD mRNAs. Next, I wanted to understand why the medium dysregulated mRNAs were so powerful. What made them similar to or set them apart from the highly dysregulated ones? To explore this, I used another machine learning technique called k-means clustering, which at the very high level is all about finding groups of data elements with similar characteristics. Imagine you're a traveling salesman and you have marked points on a map indicating potential customers you want to visit. You want to group these points into neighborhoods based on their proximity. K-means clustering places an initial set of neighborhood centers on the map, then assigns each point to the nearest center. Once all points are assigned, it adjusts the centers to minimize the average distance to assigned points, like finding the best locations for neighborhood hubs, so overall travel time starting from the hub is small. After a few rounds of repositioning, you end up with clusters of points that share similar characteristics, locations in this example. In our case, each point was one of the 100 most informative microRNAs from the HD and MD groups, described by a suite of their characteristics known as sequential attributes: length, molecular weight, the frequency of each nucleotide, and so on. I wondered if I group these mRNAs by similarity in their sequence attributes, do mixed group of medium and high dysregulation microRNAs form? They indeed did. Close to half of the resulting groups contained a blend of both highly and medium dysregulated microRNAs, evidence that at a fundamental molecular level, many of these mRNAs share structural traits regardless of their dysregulation magnitude. But maybe more importantly, there were groups populated only with medium dysregulated microRNAs, pointing to novel biochemical insight missed in prior studies. Now, let's bring this back to a likely real-life scenario. Consider the story of the overlooked patient, a 58-year-old woman with a long history of smoking. In a routine checkup, her physician ordered a standard panel focusing on the top 5 loudest mRNA biomarkers. All were observed within normal ranges, and she was sent home with no further testing. 6 months later, she returned with troubling symptoms. A follow-up scan revealed stage 2 lung cancer, detectable, but only because her symptoms had advanced. Now imagine a world where this patient's initial test included medium dysregulated microRNAs. Our model would have flagged a subtle but consistent pattern, dings from the triangle, prompting earlier red flags and intervention. Early detection could have meant treatment commencing at stage 1 of the disease, a dramatically better prognosis, less invasive treatment, and a higher quality of life. Let's revisit our concert analogy. If you tune out the triangles, the flutes, the subtle strings, you lose the nuance in the music. Yes, the brass and percussion may still roughly define the melody, but the softer instruments add indispensable depth, emotion, and color. Similarly, in our bodies, the most extreme molecular changes are easy to spot. But it's the interplay of all players, big and small, that tells the whole story. By reframing our focus from only the loudest biomarkers to a larger group, including the medium dysregulated ones, we unlock hidden layers of information. With random forests, we taught a decision tree to listen to these subtler signals, and they performed just as well. With K-means, we discovered that many of these mRNAs share the same or similar structural characteristics, suggesting common or closely related biological roles. Through that same analysis, we identified groups of MD mRNAs with unique characteristics pointing to novel insights. And for the overlooked patient I mentioned earlier, this shift can translate to earlier detection, less aggressive treatments, and a life saved. So, to all of you, scientists, clinicians, engineers, artists, students, and thinkers of every stripe, I invite you to reimagine your world. Is there something you've dismissed as unimportant that could be worth a second look? What have you been ignoring because it seemed insignificant? What data have you dismissed because it whispered instead of shouted? By embracing those subtler voices, we may find breakthroughs that were hiding in plain sight. Imagine a future where earlier disease detection is possible and more accurate, where therapies are tailored based on a richer tapestry of biomarkers, and where every piece of biological data, big or small, plays its part in saving lives. That future starts when we care to listen to the triangles. Thank you.